Welcome everyone to our basically next series of Kipoi seminar for this year and then the year 2024. And we're very happy to kick it off with David Kelley, who most of you probably know, but those of you who don't, so David is a group leader at Calico and interested in basically deciphering gene regulation using machine learning models, and he's been very useful to our community by providing a lot of different dog-named models. So people probably know Basenji, Bassett. Then there was Informer, which is kind of a bit of an outlier. And now there's a very cool new model, Borzoi, which basically works on predicting RNA-seq data. And yeah, I guess you'll talk about this a lot to us in this talk. And as David says, feel free to ask questions throughout the talk, so you don't have to wait until the end. So we want to keep it kind of interactive. And with this, I'm very happy to give you this stage, David.
Thank you. Thank you for the kind introduction. If you believe Informer should have been named after a dog, you can take that up with Zika, as I made the case briefly, but you won out.
You won that.
Okay. So thanks so much for the opportunity to talk to this group. I love attending these. They're kind of early for me, so sometimes I just listen while I walk the dog, but you know, I'm still listening. So we've been working, kind of continuing on this trajectory that we have been. Our North Star is to be able to really effectively predict gene expression as well as other sorts of attributes related to gene regulation from the DNA sequence, and then put them to work on biological problems that we care about. So, you know, there's a couple routes in here. I think there's a compelling argument that these methods can help you learn regulatory grammars. I'm going to take a different route in, one that connects a little bit more with Calico and the mission that we have there at the company, which is to lean on these models to be able to predict the effects of genetic variation. So the problem, as we see it, or the opportunity, is that genome-wide association studies for common diseases where there's an aging component are the ones that we're most interested in. Here I'm highlighting eGFR measurements in the kidney, which is a major component of a chronic kidney disease diagnosis. That's just an example of a recent effective large-scale GWAS where there's 878 different independent associations. And as many of you know, these association Manhattan plots are sometimes optimistically labeled with the genes. Here's a common scenario where the linkage disequilibrium between the genetic variants means you're not really sure what the causal variant is. You're definitely not sure what the target gene that you ought to be really thinking about that's implicated by the association is. And you also don't know, you know, in the case where it's non-coding, in the case where it's affecting gene regulation, you also want to know, like, what moment in time, in what cell is this most relevant to driving this association? So these are the goals we have. We want to precisely identify those causal variants and figure out how they affect phenotypes. And given that a lot of these are non-coding, in that case, more specifically, what genes affected, what cell type situations, which we, I think, have the data to answer now with all of the single cell atlas work and people profiling really specific cell type states in disease. You know, think of all the papers you've seen with people doing single cell RNA-seq in the immune system and all the subtle immune states that you can find. So it's not just type. I think it's also like really nailing that specific situation. Anyway, so our approach to tackle this problem is, of course, to train sequence-based machine learning models, as this group, I think, deeply believes in. And, you know, I'll highlight just briefly our previous attempt at this, the Informer model, just to kind of briefly explain the overall framework here. we're going to be collecting all sorts of potentially relevant functional genomics data from the big databases for human. And what I've learned over the few years working on this problem is that it kind of helps to have more species. So we also grab mouse data too. And we train these deep learning models to predict those data from the underlying sequence. And in the informer model, we're using convolutions to do the initial recognition of sequence patterns like transcription factor motifs, to start to synthesize regulatory elements like promoters and enhancers by continuing to do convolutions that are taking advantage of this locality inductive bias. Initially, a TF binding site is really just a k-mer that the transcription factor likes to bind more than random k-mers. And then a regulatory element is, the right combination of those motifs. So the convolutions, I think, are sufficient for this. Here's a claim I'll make that someone can debate. I don't think we need transformers down to nucleotide resolution, even though some people are starting to publish things like that. But at some point, I think we do need something like a self-attention, because once you've synthesized those local regulatory elements, you really want to figure out how they're interconnected on a much larger scale. So in the informer model, we were using 200 kb of sequence, which is enough to cover a lot of, but not all, enhancers that might be regulating the promoters. And the self-attention, we've maybe heard it before. I don't have slides on this actually, but I probably have already presented my slides to this group. It just gives every two positions in the sequence, the opportunity to interact and exchange information. And so a promoter can identify all potentially relevant enhancers around, and then the vector representation of that promoter can be updated according to those enhancers. And it goes both ways, so all the enhancers are affected by all the other regulatory elements. I think it's a really nice framework for what's happening at that stage of resolution in the genome. And that came through with our ability to make good predictions with this informer model, where we're predicting lots of different data sets, DNase data sets, histone modification ChIP-seqs, transcription factor ChIP-seqs, and CAGE, which was our kind of the best gene expression proxy that was readily available, because it marks these promoters where transcription is starting and where most of the information to predict the tissues in which that gene is expressed and the level of expression, most of the information is there at the promoter. Okay, so I spent a little extra time on those assays to try to pull the question out from the crowd. Why aren't you predicting RNA-seq? RNA-seq is the most commonly used genomics assay. If you go, look up at Geo and you kind of sort by all the different assays. I don't know, I haven't done this, but my guess is there's probably 10 times more RNA-seq than anything else. So the reason we haven't done it previously with Basenji or Informer is just that RNA-seq felt too complicated. So the coverage pattern from RNA-seq is covering the entire transcript. The transcripts can be very large. There's lots of genes that span hundreds of thousands of nucleotides, and the coverage on something like a 3' UTR for one of these long-spanning genes could be very high. So your loss function of your model really wants to get that right, but the sequence signals that determine that expression can be very far away. In that case, maybe the promoters 200 or 300 kb away, enhancers that are upstream or even farther. So it just didn't seem like it would work. For it to work, you'd have to learn transcriptional regulation, splicing regulation, and termination polyadenylation in one model. It just sounds, couldn't be done. However, I've kind of been keeping this in the back of my head that there are a lot of really compelling reasons to want to do this, one of which I've highlighted already. There's an enormous amount of RNA-seq data, and it spans way more species. There's a bunch of species where we've done some RNA-seq experiments. We don't have like an attack-seq or chip-seq atlas, which is what the informer would need to take advantage of that species. So that's kind of a missed opportunity. And a lot of the single-cell genomics, the single-cell attack-seq is super. People are working really well with that. So single-cell RNA-seq is much harder to work into this framework of predicting from sequence. So, that is a missed opportunity, I think. And then there's a lot of post-transcriptional profiling assays that read out with RNA-seq. There's ClipSeqs to do RBP binding, and when you do AGO, the microRNA core complexes, you can profile microRNA binding. When we measure RNA half-life, we read out with RNA-seq, RNA methylation, structural profiling. All of these are ultimately a fancy set of steps followed by RNA-seq. And so if you could work with RNA-seq, you could you'd have a kind of a simpler path to building models for these types of things. You can build models for these types of things using the process data, but I would argue that has some limitations. And then, this idea that you'd be bringing multiple layers of regulation into one model, it has some perks. It has some reasons you would want to do that. You do see lots of interaction between these layers. Co-transcriptional splicing is one, meaning that like as the RNA is being transcribed, it's typically also being spliced. You're like splicing out the first exons while transcription is still happening. There's a bunch of little examples like that. Okay, so hopefully you're on board. We want to do this. So, we got a whiff that this was possible. We actually have some unpublished work. I'm not sure if it will see the light of day, hopefully in C. elegans. We have a collaborator at Calico who's working in C. elegans, and C. elegans has smaller gene spans. It's a much more gene-rich, gene-dense genome. And I was training models and playing around and started seeing things like even the models that weren't working with RNA-seq were learning splice sites. And of course, we were influenced by the Splice AI, a dramatically successful splice site predictor. So clearly, there was not a lot of that. Splicing is a learnable characteristic of sequence. And lots of folks, including my collaborator on this project, Johannes Linder, have been training models on polyadenylation. So in isolation, all these things felt tractable. It's just really, how do we bring them together? And the C. elegans with the denser genes is where I first actually tried this and saw that, oh, actually this works. Guess I'd been kind of underestimating my deep neural networks, which you wouldn't think would be true after all these years of amazing successes in the field, but here we are again, finding new capabilities. So we jumped up to human and mouse. The big jump there from C. elegans was that ultimately the gene spans are much larger. So the difficulty of like stitching together all of the exons and kind of calibrating the gene's expression is harder. Figuring out the start side and the end side across very large distance is harder. But the self-attention mechanism that we demonstrated with the informer felt like it was up for the job. The one thing we wanted to tweak, there's a few tweaks in this new model relative to informer, but one of the biggest ones is we really wanted to predict at finer resolution because 128 base pair bins felt too big. There's a lot of exons that would then just kind of fit in that bin, and then you're not really getting meaningful predictions on the splice junctions. So we wanted to shrink that bin size a little bit. But actually so the simplest way would be to just do fewer layers of convolutions to something like 32 base pairs, and then do self-attention at 32 base pairs. And then, it just looks like the informer, but you never went to 128, you just stopped at 32. But the self-attention mechanism that we use, because of this complicated relative positional encoding scheme, has quadratic memory complexity. So the sequence length at 32 base pairs And by the way, on the topic of sequence length, we doubled it relative to Informer because we needed to fit a greater proportion of the gene spans into the sequences. The memory complexity is just too big to do that. We want to do that. There's a lot of techniques in the ML literature about how to do self-attention efficiently without quadratic complexity. We are actively exploring these. I'll take full responsibility, a bad decision several years ago to make use of TensorFlow for all our programs. No offense to TensorFlow or my employer Alphabet, but all of the new techniques that people are exploring in the ML literature are publishing code in PyTorch and JAX, and it's really a pain in the butt to have to port them all to TensorFlow. Anyway, we'll crack this at some point, and we'll have more efficient self-attentions in these models, or someone will beat us to it, and we'll borrow from them. But for now, what we did was steal a technique from the image segmentation and object detection literature, where when you want to solve those problems, you want to do your whatever deep learning block convolutions, or some people just do self-attention now, up and get the whole sense of the whole image, what's going on in that image. But then you want to zoom back down to the pixel level. So actually, I'll just use this image here of a dog called a Borzoi. It's a beautiful dog breed. So you want your model to get a sense of, like, this is a dog, it's a Borzoi, it's on a dirt road, it's in a park, there's a sunset, park, maybe farm, something like that. So now you have an overall sense. So you have a vector representation at the highest level that captures all those things. Now you want to zoom back in and say, these pixels are the dog's hair, this is the dog's nose, this is grass in the background, so on and so forth. So the way people often do this is using what's called a U-net architecture. And the idea is basically that you save these representations in your initial tower of convolutions, and then you pass them along. So the one path goes through these transformer blocks and gets the whole global sense of what's going on. So if I'm a promoter, now I see all the enhancers. If I'm a middle exon, now I see all the surrounding exons. But then when you want to solve this problem of, okay, I'm at 128 base pairs, I want to get down to 32, what should my prediction be in each of these bins? Now you want to bring in the underlying sequence and what we originally learned in those convolution towers back in, and then just do a little bit of neural network magic to combine those things. And now you can say things like, all right, at 120 base pairs, I know there's an exon here, it's a small exon, but then at 32 base pairs, I see the donor site, acceptor site, and I can kind of partition out the coverage properly. I don't know if it's doing anything much more super sophisticated than that, but it seems to provide some value. Okay, that was my longest presentation of this architecture so far. So let me pause, and I see there's one question. We predict RNA-seq at different base resolutions. Is the model guided to predict at the same time? No. We don't do a multi-resolution thing. We just do the 32 base pairs. I don't have a lot of intuition for whether predicting at multiple kind of nearby resolutions would be super valuable, but I don't see any reason why you couldn't do that if you did see a reason that it would be valuable. Okay. Wouldn't this help in reconstruction accuracy? Maybe. I'm not sure. Just, yeah, I don't have the intuition for that. Okay, moving on. Oh, one more. Did you see any impact of using different normalization schemes? Okay, yes, good point. The data is super important. We really didn't spend a lot of time with this. So we kind of subsequently spent more time with this. The major source of all this data is from ENCODE. That's just the easiest thing to do these days. to download all the bigwigs from the ENCODE portal. So the first thing we did is grab all the RNA-seq data sets there. The coverage tracks there are alignments from star, and then there's a mode where you can run the star aligner, where it's giving you back effectively a normalized coverage track. That's what we're predicting. So the tracks are to some degree normalized by star, but they're all kind of run in isolation. There's no like sophisticated cross-track normalization scheme happening. I kind of go back and forth on whether that's a good idea or not historically with the rest of the data too. You know, for the cage data, for example, I didn't care for their processing, so I processed all that myself. And so I'm just using aligned read coverage tracks for all the cage. And I don't normalize it because the data sets do have different depths, and the depths are a pretty good proxy for the quality of the data set. And we use a Poisson loss function. I've just always found that works better than some sort of mean squared error thing. And the Poisson kind of naturally handles these differences in depth, where it's going to have greater magnitude gradients coming back, and it's going to make more use of the higher depth data sets. And so I think there's some benefit to not normalizing and just saying like, okay, here's a high depth sample, you know, capture that with the Poisson loss, and here's a low depth experiment, and maybe you want to consider that less. But it's an open question, what would be best? And it is definitely a little inconvenient at the end when the model predictions are not normalized, because you then say you want to do variant effect prediction, and you have a high depth data set and a low depth data set, and the values are bigger for the high depth data set, and you have to like figure out how to calibrate that back out. So I would say this is something we're still kind of actively trying to think about. It's also worth noting that we don't just let it rip with these coverage tracks. RNA-seq is typically studied on a log scale once people go to gene expression matrices. Log felt a little dramatic to me to introduce to these coverage track profiles. And so what we ended up with was a admittedly kind of complicated scheme that's based on exponentiating to values less than 1, which introduce a squashing effect. I'm hoping we can simplify that in future iterations, but we are introducing basically like a squashing function to get away from being, you know, completely dominated by predicting albumin gene expression, which would be like orders of magnitude larger RNA-seq coverage tracks in the liver, for example. So, you know, that's not a normalization per se, but it is like a transformation of the original tracks. And Julian's encouraging us to keep going. Okay, we'll come back to some of these other questions. All right, so, you know, overall, let's start looking at some results from the model. We've described the model. We can talk more about it later, but here's a gene that I picked out. I kind of sorted a table of genes by complexity of their intron structure and gene span, and picked one I recognized. And, you know, this one is qualitatively predicted very well. This is a gene from our test set, so the model hasn't seen this before. And it's pretty easy to tell scrolling around through the browser of these expression predictions that, you know, at a coarse level, the model is definitely learning the splicing structure, the exon intron structure of these genes, and roughly predicting calibrated gene expression levels, and then also figuring out where the genes stop too. So on the bottom left, this is just a bin. Oh, no, both of these are gene level, sorry. So for the gene level, if we want to think about predictions at the gene level, we just take the gene annotation quick note, there is no gene annotation in the modeling stage. It's just sequence bins. Every aspect of where a gene is, where the exons are, the starts and ends, the model has to learn all of that de novo. That's another modeling decision that, we made, that you could potentially try something different. Splice AI, for example, is training on the annotation. That's interesting. You could do that here. You could train on the annotation and all this RNA-seq data. That might be nice, but we just found it wasn't totally necessary because the model's pretty good at just learning the gene annotation without having ever seen it. In the bottom right, we're, okay, so wait, sorry, skipped ahead. So to think about gene expression, we then bring in the annotation and we sum up expression on the gene and we kind of undo all the transformations to get to something that should be a good proxy for like the log TPM value of that gene. It's a little tricky, when we plot these things versus real log TPMs, they're not on a line, they're sort of like a little bendy, but it's not bad. And then we can put the data itself through the same transformation and then compute accuracies. But what people tend to care more about than the overall level of gene expression as like a absolute level is the specificity across all the tissues and cell types. So on the right, that's what you're seeing. I'm subtracting out the mean. First, I'm normalizing more carefully because I have a gene expression matrix now. And then we're subtracting out the mean and computing accuracy too. So for each gene, you have this vector of like, I'm higher in the liver and I'm lower in the brain and a bunch of other tissues. And you have to have a good correlation between those vectors. And so you can see we're, imperfect, but good. Okay. Okay, so Johannes put together this wonderful visualization that I'm not sure everyone who reads the paper is going to dig into deeply, but if there's anyone, it's this group. So this is a self-attention matrix, or it's an aggregation of numerous self-attention matrices. Johannes can correct me if I make any mistakes on that, for a more or less random region that had some genes. And we thought this was a neat view into how the model's actually working. So you can see these vertical stripes are basically the most interesting kind of regulatory elements of this region. And the vertical stripe means that lots of different places around are paying attention to that location. So each of the rows is, for that position, what is it attending to? So if you slice across a row, you see, I'm paying attention here, 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 so on and so forth. Then if you go within genes, so basically there's a lot of cross-gene communication, I think is one note, and it's mostly at the boundaries of the genes. The promoters are all looking at the promoters and everything's looking at the promoters. The promoters are pretty important. Lots of things are looking at the exons. Lots of things are looking at the end sites of genes. If you zoom into a gene, then, all the exons have to attend to each other because they have to calibrate their expression. There's like a determination of the overall expression level of the gene, and then that has to be sort of distributed to all the exons, and the self-attention mechanism is the primary way in which the model is able to do that. And then if you dig in on the far right for where the genes ends, you see different polyadenylation signals are attending to each other, as well as the splice sites nearby, because there's some interaction between these things. And for all of these different features that you can derive from the attention maps, you can establish that as a numeric value and compute gradients back to the original sequence to see how it changes as a function of the sequence. And, we see that things like attention on promoter is determined by the TF motifs, attention on splice sites is determined by the donor acceptor sites, and similarly with the polyadenylation. So I think, there's a lot more that could be done here in terms of interpretation, but here's a cursory view so that we can keep going into how the model is making predictions and establishing really long-range relationships. Okay, So big question we often get asked is, you get the same DNA sequence in every cell, but you're making different predictions in each of those cells for gene expression or attack-seq or whatever. How does your model do that? Usually these people have like a vague notion of epigenetics in mind. And so here's my go-to slide now for addressing this question. So what Johannes did here was start to really dig into gradients from the model. So when you have such a large region of sequence, the ISM type methods kind of break down. It's hard to do ISM at a 500 kb sequence. I know there's some folks in this field who've been publishing techniques to try to scale these things. We're quite interested in that. But for now, the easiest thing to do is to choose a subset of the tasks that you're most interested in and use gradients to do a backpass for each of those tracks back to the sequence. And then in one backpass, you get, all the gradients for this really large sequence. So we did that for a lot of genes that are differentially expressed in a bunch of GTEx tissues. On the left, upper left, you could see an example of that for a liver-specific gene, where the gradients highlight TF motifs that are specific to the liver, and this gene is not expressed in blood, brain, muscle, esophagus, so you really don't see too much action there. So for these little motifs, we can match them to the database, but we can also do a de novo clustering. We use the TF Modisco technique from Anshul Kandaji's group to do that. And when we do that for all the different tissues, if you move over to the right, we find that we're able to kind of de novo discover from RNA-seq gradients all of these different fairly well-established TF motifs that are specific to these different tissues. And, below the motif, we're plotting a kind of swarm plot-ish thing, box plot-ish thing that shows you the full distribution of the gradient scores for that motif in the different tissues. Something that was interesting is, there's a lot of use of these motifs across the tissues, and probably the more interesting one was the CEBPA/B, where it's both active in the blood and the liver. And the thing we noticed here, is highlighted by this red-blue scatter plot, is that those are tending to be different instances of the motif. It's not the same genomic locations that have high gradient scores in blood and liver for the most part. There's a couple of those, but there's way more that are either blood specific or liver specific. And, I'm going to take a guess at why that is. I don't have super strong quantitative evidence, but, I think it's largely determined by the other motifs that are around and whether you have enough of a regulatory element forming for liver versus blood, and the CBPA may be a little more of like unable to function alone, but needs to function in the context of some other TF motifs nearby, and that's where you get these tissue-specific components. Supporting the validity of the specificity of those motifs is that we actually do, because this is RNA-seq, have RNA-seq measurements for the transcription factor itself. And, there's some ambiguity with these families, and, the CBPA versus B, those are two different genes. They have two different TPM values in the RNA-seq, so we have to do a little aggregation thing. But when you do that and you look at pairwise comparisons, you see that the gradients for the motif are strongly correlated with the differential expression of the transcription factor. So that's a good sign. When the TF is more expressed, the motif is more active. That's what's driving the tissue-specific predictions made by the model. Okay. So one of the major evaluations that we've picked up for the last couple attempts at this problem is to work with this kind of medium throughput CRISPR-based perturbation of enhancer elements, followed by a gene expression profile and kind of a rating of how that enhancer behaves relative to the gene. There's two different big experiment sets, one in which teams led by, Jesse and Grades 1, and there's a bunch of others too, have gone gene by gene and developed assays and measured a whole bunch of regulatory elements around those specific genes of interest. But there's also a single cell based whole transcriptome screen from Jay Shanduri's group where you target regulatory elements all over the genome and you use RNA-seq to read out the gene's expression as well as the perturbation. It's kind of pretty neat design. So the data looks like this. Here's two examples for two different genes. All of the vertical bars are places where the CRISPR machinery was targeted to that regulatory element, and there was a call made using the output of the experiment on whether it was significantly affecting the gene's expression or not. The reds are not, the greens are yes, and the track I'm showing you is us taking gradients back to the sequence, but then running a smoother over it. And so coming to a resolution is kind of like, it would be as if we were roughly averaging in 1,000-ish nucleotide window so that we get a kind of nice smooth signal that scores regulatory elements rather than sequences or motifs, so rather than nucleotides or motifs, because that's about the resolution that this CRISPR is actually targeting. So the top one, HBE1 here, is a real victory for us. Most of the red lines have low scores, although a bunch of them do have little pop-ups. And the main enhancers for this gene are in this like little window. I think it's something like 20-30 kb, where we have high gradient scores for each of those. And there's multiple lines here. One thing I didn't note, but I should talk about with this group, is that we trained 4 models this time. With the Informer, we trained a bunch of models in development, but then we released and studied 1 model. In this paper, we studied 4 the whole time. In any case we could, we used all 4 and we ensembled them. But, it's very useful to kind of keep them apart in many cases. You can see a little bit of variation of how the 4 different replicate models make predictions for these things. And you always get better results by averaging the 4. Almost always. Another example down here is MIC, and this one is a little more of a mixed bag. There are definitely some false positives in there, but the one victory is this really distant enhancer that we then did a visualization of the gradients of the sequence, and it's not too surprising that this is a K562 enhancer. K562 is addicted to the transcription factor GATA, and this is a GATA enhancer. So that's good. At the bottom here, we're just showing kind of whole transcriptome regulome across all the data that each of these 2 different studies introduces, and binning the sites and showing that our ability to classify the positives from the negatives is better than the Informer model. Okay, let's come back to genetic variation. And I see the time. I want to make sure I leave some time for questions too. So, maybe we'll fly a little quickly through the eQTLs, because I think everybody's seen eQTLs before. This is just another example. We're studying an eQTL. The model makes a good prediction. Using these interpretation tools, you can tell why the minor allele C introduces a motif for the CEBP factor, and the whole regulatory element kind of rises up. This IRF1 motif and the SPI1 motif, they're there in the reference version of this sequence. There's just not enough else interesting going on. And it's this third motif that really pulls it up, makes it an enhancer. And the really neat thing about Borzoi, the new model, is that you can make predictions for the whole coverage track of the gene. We can see lots of finer resolution things that are happening, but often it's just simpler to aggregate back to that gene level. And in this case, this is one we get right. If we look at eQTLs across all the GTEx tissues, and we compare to our ability to predict those to Informer, we have better predictions for all the different tissues, except maybe this one weirdo tissue. Just quickly to note, there's a couple iterations of this in the paper. One thing we realized was we were under the score we used in the Informer paper was not optimal. We were just looking at the sum of one version of the allele versus the other, the sum of the coverage. And actually, there were cases where it goes a little up, a little down. So what we like to do now is take the two tracks for the reference and alternative, subtract them, and then compute a L2 norm. So you're looking for like shifts throughout that are introduced by the genetic variant. Okay, I also want to briefly talk about how these models are not currently great at predicting gene expression across individuals in a population. Basically, if we come back to this, we're making good progress on predicting eQTLs, but we still got a 0.8 AUROC. That means you're making a lot of mistakes. There's a lot of things that aren't eQTLs that your model thinks are. There's a lot of things that are eQTLs that your model thinks aren't. There's also a lot of cases where your model thinks it's an eQTL and just kind of misunderstands how it's affecting that gene. And these two preprints are highlighting those cases, which really burn you compared to these models that people use for transcriptome-wide association studies that are basically just regularized regression models on the genetic variants themselves. So for any common variant, these models have maybe 50 examples of each allele. At that point, you have the right answer. Like, there's no machine learning model that is going to beat that. You've just gone out and measured the thing, and like that's ground truth at that point. So, I think this is a super important problem. We're kind of adding it to our suite of benchmarks. We need to get better. Here's where we're at. On the right, you can see for whole blood, this is our prediction of the eQTL effect size versus the measured eQTL effect size. And, these correlations are going up relative to Informer. I hope they'll go up again if we introduce a new model in the future, but there's still too much happening on these off-diagonal, the bottom right and upper left quadrants. So, to all who may want to continue to tackle this problem, I would say that the best path forward is not to completely lean on the machine learning model, but find a clever way to combine the machine learning model with what folks are already doing. Okay. I see question in the chat. Was the ensemble or a single model compared against Informer? A little bit of both. We did a little bit of both. A single model is better than Informer, but an ensemble is better than both. Individual genetic variation in the model. Let's talk about that at the end. That's much, much, much more complicated. Okay, so in addition to just predicting whether something is an eQTL, there's also different types of molecular QTLs that people call from RNA-seq. And one of them is focused on polyadenylation. Before we get into the QTL, let's just ask the question, does the model correctly predict these polyadenylation transcript termination sites? So if we look at genes where there's multiple sites, for example, this one, there's a distal and a couple proximal sites, and we can compute a ratio of the coverage predicted at the distal versus proximal. And in these scatter plots, you're seeing that relative to the real data, the model predictions are very good at this. So we're very good at figuring out where the transcript ends, and if there's multiple options, kind of figuring out the relative strengths of those two different options. And when you dig into the attributions that determine this ratio, you see that the core polyadenylation hexamer is always key, but there's a bunch of other motifs that pop up that that we can cluster again with TF Modisco and annotate as being known to be involved in this process. So for the genetic variants, we tapped into a database called the eQTL catalog, where they did uniform processing of GTEx and a bunch of other eQTL data sets and called polyadenylation QTLs, splicing QTLs, transcription start site QTLs. It's a really nice database for benchmarking models of this sort, and I recommend other people check it out. So here's a couple examples of polyadenylation QTLs called from the data the kind of second row of these tracks is the RNA-seq data that we pulled from GTEx individuals who have the minor allele and major allele. And so you can see the difference between the two different alleles in the real data. And then in the top row is the model prediction after introducing this variant. And in both cases, we can then further dig in and find, using interpretation techniques, that there's some motifs that are influencing the choice of polyadenylation site. They're not the core polyadenylation hexamer. So arguably, these are ones that would be, harder to find if you, just used a very, very simple technique of, does it affect the core polyadenylation hexamer? So that's why we picked those out. Again, we can play this game we did with the EQTLs where we have, and I should say for the EQTLs, I skipped a detail. We're working with fine mapping of these EQTL associations. I showed you at the very beginning that GWASs are often very affected by linkage disequilibrium. It's not always true that you know the causal variant. We focus on the ones where we can estimate the causal variant with high probability, greater than 0.9, using a statistical fine mapping tool called SUSY usually. And so we have these fine mapped polyadenylation QTLs. We can choose a negative set that controls for simple properties like distance to the polyadenylation site and expression of the gene. And then we see that, okay, we performed pretty well at this task. Johannes wrote a parent 2. So if he says it's better than a parent 2, then, this is a unique opportunity where you get to, most of our benchmarks are versus tools that we wrote, which is hopefully compelling to the reviewers. Okay, splicing QTLs as tools that we didn't write, so you can be more skeptical of this, but I think there's a lot of reason to believe that this tool, the Borzoi tool in predicting RNA-seq, will be the right path forward for splicing. I will say that in the benchmarks we did, first we tried to play on their territory and just predict the splice sites. And the benchmarks I could set up, these programs are really good at predicting splice sites. And when you're predicting RNA-seq, you're affected by like low coverage on the exon. So we couldn't easily beat them on splice site classification, but I don't think that's quite the right, most important problem here. So we focus on the splicing QTLs, and we wanted to focus on things like tissue-specific alternative splicing, but unfortunately, there's not a lot of evidence right now that these models are very good at tissue-specific alternative splicing. We'll come back to that maybe at the end. But for the QTLs, again, we can play this game of like, let's look at a fine mapped SQTL. Let's pull the real data. Let's make the predictions. Those correspond well. We can do the interpretation and see. It's not the actual donor on this one. The donor is, or the acceptor is nearby. So we're kind of hitting near the acceptor, and there's a little track that's important. Compared to Pangolin, which I think is arguably better than Splice AI, it's definitely better for us because it has an open source license, which Splice AI does not. As a company that releases software in this space that's always open source, I think I'm in a fair place to critique them for that. So we like Pangolin, and we like it too because it performs very well at this task. It's, we kind of roughly come down as a tie versus Pangolin in predicting splicing QTLs. There's some interesting distance effects where Pangolin actually beats us at the greater distances from the splice site because it's pretty good at picking out these like gain of function splice sites. This is something we're still digging into. Having a much larger context feels like it should be the right solution to this problem, but I think it might need a little more development. Maybe the addition of some additional training data that really highlights the contributions of different splicing factors and other things that are going on before we have like a real meaningful breakthrough on this sort of stuff. Okay, and then real quick, I made the argument that unifying these layers of regulation into one model has benefits. This is, I think, the best case for that. For polyadenylation, there's this competition happening when there's splice sites towards the end of a transcript and when there's a polyadenylation signal in an intron. Basically, the transcript will stop at that polyadenylation site unless you splice out the intron. And then it's not there and you can keep going and hit some more distant polyadenylation site. So the strength of the splice sites can affect the polyadenylation here. And that's kind of what we see for these examples. We picked out some examples from the QTL data set of intronic polyadenylation, QTLs, and looked at our ability to predict them and did some interpretation. And you can clearly see this, this relationship happening. Okay, so broadly, I argued that the big reason to do this for us at Calico is to make better use of genome-wide association studies for common diseases with an aging component to them. And so a lot of the projects that we're taking on next are in this direction of making use of variant effect predictions, of which we now have a very rich set of different types of variant effect predictions. You have whether you think it's an eQTL, polyadenylation QTL, splicing QTL, and then you have that for all these different tracks that you've studied. So it's really, it's an interesting opportunity. You have a lot of information. You almost have too much information. And so figuring out like, how do we help guide these statistical genetics tools that are trying to make discoveries, determine the precise causal variants, determine the right target genes to prioritize, and then figure out what cell types and particular states of those cell types are the most relevant, are all places where we feel like we should be able to make an impact. And that's an active area of research. So a whiff of there being some value here is this experiment that many folks do in the field now, where they try to look at common variants versus rare variants in a database like Nomad. And, we can see versus generic variant pathogenicity scores like CAD that, we're on par, and CAD's kind of a meta classifier. So, if you were to combine Borzoi and CAD in a more clever way than we did, you'd probably reach even better results. So that's a good sign that we're like picking up on the right things to be able to help drive some of these objectives. Okay, so with that, we can conclude. You can kind of skip the first bar and block and focus a little more on the second, that we introduced this new model, Borzoi, to predict RNA-seq. All of the benchmarks we have for transcriptional regulation are boosted relative to Informer. We also were able to study splicing and polyadenylation at more or less state-of-the-art levels, especially when you just go ahead and combine. Since the approaches are different, there's often a value in just kind of combining them, and nothing wrong with that. And ultimately, these variant effect predictions, we're really hoping will superpower statistical genetics and help gain more insights from super big GWAS. And with that, I will thank the team that worked on this project. Big team from my group over the last few years, including Vikram Agarwal, who moved on to Sanofi, as well as the folks depicted here, Han, Johannes, and Divyanshi. And Johannes really drove a lot of the amazing figure making that you saw throughout this. And thank you all for your attention. And you can check out the preprint here if you haven't already. And okay, I left 6 minutes for questions, so I'll try to speed answer questions.